For this team meeting, I'm going to explain a bit of the most, let's say, novelty word presented in this particular paper. This paper is called the Neuronal Chromatin Landscape in Brain for Individuals with Schizophrenia. Is linked to early fetal development. So a bit of history is that we have been following some of the work made by this lab from the Panos Russos. So we have been follow these papers, I guess, if we see into this table. The first paper that we talk about it was the one in black bold, in bold later. This is from the 22. So it was to talk about the multi-omic profiling of the developing human cerebral cortex at the single cell level. And then we talk again about the work of Panos Russos working with multi-omic approaches and epigenomic data in a paper from 2024, where he presents data about genetic regulation of cell type, specific chromatin accessibility shapes, brain disease etiology. And now we have this new paper when they release this new paper, when they make an extensive work on the epigenomic side, and they present sort of novel workflow. So I will try to focus my talk into present to the team what are novelty factors in this paper that we have not discussed earlier, and also to try to present new terms that are arising related with the epigenomic analysis. So as this is a very extensive paper, I try to focus my talk in very specific, let's say, goals that we can reach in this meeting. So I will explain first a bit of the background of the epigenomic side, and then I will explain the workflow that the authors follow to identify this module of open chromatin regions that are related with the schizophrenia. So now, this is the paper that I'm presenting here. Basically, it's the same information here. Also, this is a brief of the information that I explained about the previous paper that we have studied with this author. In the first one, they profiled gene expression and chromatin accessibility in 50,000 cells in 6 broad developmental times from fetus to adult. And they, again, focus on schizophrenia and bipolar disorder. And in the second one, they measured chromatin accessibility in 2,000 aliquots of sorted neuron and non-neuron. So since this paper, they present interest to study the difference between sorted and non-sorted neurons. In this case, they do that in 616 human postmortem brains and identify 30,000 open chromatin regions with chromatin accessibility quantitative traits loci, meaning that they identify like a bunch of these open chromatin regions that are acting or regulating a bunch of genes. So now is this like sort of continuous work that gather the techniques that they used previously with a novelty technique where they identify what is called the TRDs, that is going to be most of the focus of my talk today. Then what we have next is that, I'm sorry, I will make a break here just to say that I highly recommend people interested in epigenomics and in multiomes to read this paper. This is a review from 2021, but it sets pretty well many of the terminologies and concepts that we actually use in many of the papers. So I think that it's really useful to study, and also because it's focused on disease of human brain. Then, saying that, I will just use 5 minutes to explain a bit of the concepts that we actually know about of the chromatin. So this paper that I'm presenting use a combination of different epigenomic protocols. Most of the paper use ATAC-seq, but it's not the only protocol to measure chromatin. ATAC-seq has the particularity that it focuses on to read open chromatin regions. So if we focus on this cartoon, we can see that we have in the nucleus the chromosomes, but the chromosomes can be present in two states, basically. One is called euchromatic, and it's what we call the relative open and accessibility region that we assume that has transcription start site that is going to act into the regulation of genes. And also let's say the other state is the state that we consider like close. So that is here depicted in these three nucleosomes that we are looking at here. That is a state called heterochromatic, and we cannot have access here to the transcription start site, so the regulation is basically silenced. And so this is like the fundamentals of the way that this transposasa works in the ATAC-seq protocol. Next, we need to remember that basically the transposasa that takes these fragments in the open chromatin are in let's say the most common interpretation is that when you have an open chromatin region open, so you have here possible transcription factor that can bind an RNA polymerase that is associated with the DNA of coding region. So let's say that this is the active region when we are expecting to have transcription. In the other side, when you don't have access to this, it's like when you have some kind of block impeding to be binded for activators in order to activate the transcription. So this is called in general repressors. So in most of the talks on the papers, we are going to find these related with activators and repressors or similar ways to describe the same. The other concept that is going to be focused on this talk is that also the genome and the open chromatin regions fall through they fall and they have like different regions of open chromatin, and these different regions of open chromatin is synthesized that they can act together in order to activate a lot of genes at the same time. So this is to say that even when we define an open chromatin region to measure genes that can be activated for a specific transcription start sites or transcription factors, also exists the possibility that there are distal open chromatin regions that are acting together to co-express a bunch of genes at the same time. And this is under the hypothesis that many diseases are not only driven by one, two, or three loci or specific genes, but they are driven by the bunch of genes. So in these papers, the authors focus on workflow to tag or to try to identify these modules of open chromatin regions that could be acting in schizophrenia. So another terminology that it's necessary to have clear before to explain the paper is that there are different kinds of regulatory elements. These regulatory elements usually are described based on the distance that they have from the transcription start site. So for example, if you look at this line is simulating a genome. And in the genome, we have identified a transcription start site, and this transcription start site, depending on the distance to the different non-coding regulators that we have in the open area, can be described as promoters that are very close or very near to the TSS, contains a data box, and they have a specific like base pairs for the initiation of the transcription, and they are usually under a specific distance. But also we have enhancers, and these enhancers are thinking to be distant elements that boost or act together with the promoters. And in this case, the authors of this paper, they focus on to identify and describe this collection of enhancers that are acting to drive schizophrenia, or that they present susceptibility to have this kind of disease. Okay, so now starting the paper, Some of the key findings in this paper, I'm going to explain basically the technique more than the biological interpretation of the results. So it's important to say this since the beginning. So, for example, I only list 4 of these. This work is driven into study neuronal specific versus non-neuronal changes. The authors hypothesize that the risk to this disease, to the schizophrenia, should be driven or should be more contained in neuronal specific cells than in non-neuronal cells. Also, they identify many open chromatin regions in these both kind of cells that they classify this way, and they made a classification to or a subclassification to split it in upregulated, saying that it's more accessible, and downregulated, meaning that it's less accessible. And they compare this in cases with schizophrenia, but also they use bipolar disorder and other diseases, but as they don't have enough statistical strength to continue with the analysis, they only focus on schizophrenia risk. And then they try into this particular brain disease, they try to follow the analysis to try to identify if there is a link between the fetal and the adult state that is linked to the disease. So this is, I think, the most important motivation on this paper. And all that is focused on a new concept that I'm going to introduce. I have not introduced it before, that is called trans-regulated domains and cell type specificity. So we are going to focus a lot in this particular terminology that is used to describe these modules containing a lot of OCRs linked to the risk of schizophrenia. And well, this is more of the, well, they have resource that they create to follow the talk. Okay, I'm going to skip this because, well, no, this contains relevant information. I think that this is the more relevant. They use around 1,393 chromatin accessibility libraries, meaning that they are soluble data from neuron and non-neuron of 2 neurotypical regions. They studied the DLPFC and the ACC, and they are coming for a total of 469 donor brains. And here's a video of the technique used to separate the nuclei by neon status. And also they explain that they use as method of heritability, the LD score regression. Okay, so I'm going to explain the first figure. I'm going to use the labels of the figures coming from the paper. Just I want to, before to explain the figure, I want to explain here the difference between cis-regulator, trans-regulator domain, and polygenic risk scores, because they are used in all the figures. So here, what we have is that we have explained before what is a cis-regulatory domain. So they use basically an open window to identify these open chromatin regions that are differentially accessible between different cells. So in this paper, it's labeled as CRD, but in other papers, it's labeled OCRs or cis RDs. I mean, they have like different nomenclatures, but let's say the regular meaning is that they are open regions that are differentially accessibility by different kind of cells or depending on the classes made by the author. But this is the relevant one in this paper, that is the trans-regulatory domain. So what is that? This is called as a functional co-regulated cluster of peaks. So here we identify first the peaks, and then we need to apply a technique to identify these modules of co-regulated cluster of peaks. They are not genomic locus because, for example, to identify the CRDs, they are coming from the same chromosome. But in this case, in the TRDs, they can be in different chromosomes, not necessarily in the same one. So this cannot be measured by a a specific distance because they cross the physical organization of the chromosomes. So that is why they use a sort of weights to identify these co-regulated peaks that are acting in specific cells. And then they also talk about the polygenetic risk scores. That is the technique that they use to identify, let's say, a continuous or a kind of identifications of these clusters in relation with the risk to schizophrenia from fetus to adult. Now, some of the properties that have these TRDs is that they covary together across individuals or samples if you are using pseudobulk. They also behave as a coordinated regulatory module, so it is described as a whole, not like as individual parts, and are not physically adjacent in the genome. So that is why we cannot call them trans or cis, and are upregulated together in schizophrenia neurons in this particular paper and share common transcription factors. I mean, they need validations to just confirm that these genes are co-regulated by the same kind of transcription factors, motif, of specific regulatory programs. Now, going back to the figure, I'm sorry, I will try to make this a bit bigger. Okay, so in this panel, the first figure, I'm going to explain basically three figures from here. The figures that describe how they found the TRDs. This is the goal of this talk. So in these four figures, they describe the population scale chromatin accessibility analysis in the human brain. Basically, they divide the panel A in three sections. You can see here the sections. This is the first one in this square. The second one is all these squares that you see here that is divided by these dashed lines, and is the analytical workflow. And then they have B and C as the results that they found using this technique. So this is the most relevant because in the first one, they are describing the data generation. Basically, I have described this, the number of donors used, the number of brain areas included in the experiment, and what is more relevant is that they are looking for differences in the regulatory patterns between no neurons that are in blue and neurons that are in red. And then in this table, they are described the number of libraries included. That is a total of 1,393 ATAC-seq libraries. They are not talking about number of cells at all in all the article. They are talking about samples, they are pseudobulk data. And then in the analytical workflow, they say, okay, first we need to identify OCRs or open chromatin regions that are in schizophrenia and in bipolar disease versus the controls that we have. So basically they classify this, they found this with the regular techniques, they QC the data, they made the differential accessibility, and then they are here showing a cartoon just to describe that they could identify downregulated and upregulated OCRs in schizophrenia. And in the next one, in the next panel, they are working in the inference of the transition between the schizophrenia and the controls. So basically, this is a diffusion map. When each one of these dots that we are looking into this plot are samples. So if we look at here, we have a shift between the controls and the samples of the treatments of the schizophrenia, when we can see the transition from black to red. So what they do is that when they have this, let's say, confirmation that there is a shift between the controls and the schizophrenia, so they take this and build in the next panel a kind of network only using the OCRs that are included in schizophrenia in order to build modules of co-expressed OCRs and identify these specific modules. So here what they are doing is depicting that they have the ability to identify these TRDs, that are modules of OCRs, that are specifically related with the schizophrenia. And then they take this into the polygenetic risk scores in order to see if they have like let's say a latent scheme of the function here. So this is, let's say, all the story of the talk described here in the data generation and the analytical workflow. And they here in the panel B and C, they provide a bit of stats about of the overlappings that they have in the different kind of designs that they use. For example, the Yakar score between neurons and non-neurons, that are the cell types that they describe in the articles, but also they have the Yakar score between the two different regions that they use, the ACC and the PFC, for neurons and for non-neurons. So you can see here that the overlapping between neurons is higher than the one that is expected in non-neuronal populations. And then in the panel C, what they have is a schematic to show the enhancer to promoter links. That this is very relevant because they were really interested in to identify where they have more links related in the different kind of populations that they are used, that there are neurons and non-neurons, because they were hypothesized that the neurons are more driven by this kind of disease. I think that even when they have these numbers here, we cannot really appreciate here in these pie charts the intensity of the color that they use to say that there are more links overlapping, because it looks like pretty similar. I mean, the density of the red and the density of the color blue, it looks pretty similar. But the point here that they say that they found more intensity or more relations between enhancers and promoters in neurons. This is like the big picture of the previous results. And then what we have here, okay, this is very relevant because essentially what the author are doing is saying to the people that the TRD is essentially a network defined regulatory module inferred from covariance of open chromatin regions across all the samples. So here I extract the 5, let's say it could be more, but the 5 principal steps using to identify these modules of OCRs. So basically, it's a correlation matrix of accessibility between peaks and samples. And then they have a network analysis based in agent modules that I think is the most used approach to make this kind of modules in genes or in open chromatin regions or any kind of molecules. Also, they cluster methods to group these OCRs, and they filtered from neuronal to non-neuronal modules. They were highly interested in to look into these differences. And then they use visualizations as regulatory domains across the whole genome. So the best representation to do this is through this one. So they have all other figures when they are showing the total TRDs identified that I'm going to show in the next figure. But basically, here the relevant point is a TRD1 looks like this one that we are looking here in the image, that is composed for the correlation between these open chromatin regions, 1, 2, 3, for example, and here 4, 5, 6, that they have a similar score, and this is called, they label as the domain 1. So what follows here is that this, for example, this TRD1 is neuronal specific module that is composed for a total of 1,000 of open chromatin regions. And they found where this is important is because they found that this specific collection of open chromatin regions identified in this module are highly enriched in fetal brain, and also show motive for developmental transcription factors and also mapped to genes expressed in immature glutamanergic neurons. And also they correlate with these pseudotypes that are more accessible in schizophrenia profiles. And the point of this is this schizophrenia chromatin dysregulation, they are saying, is global more than local because it crosses different chromosome open chromatin regions. It's not isolated or individual peaks. And the dysregulation suggests that there exists a correlated regulatory program that is not randomly identified. And well, also what is relevant here is to have a clear definition used by these authors here in the paper when we can identify that the location of an open chromatin region is always in the same locus and chromosome, while in the TRDs, there are peaks across many chromosomes. And the mechanisms that they act is that OCRs, they are local enhancer promoter controls, while in the TRDs, they coordinate co-regulation across genome. The scale also varies from tens in 100 kilobases to tens of thousands of peaks. And the biology or the interpretation in the biology context is that OCRs are gene-specific regulation, while TRDs are always interpreted in the context of global regulation programs. Okay. And then the next point is the general polygenetic risk scores. That this here basically is the third part of the workflow panel that they show in the figure 1. And let's say the steps followed for do that is to select the SNPs that fall only in the schizophrenia-associated neuronal regions in the module TRD1. So this is a very particular feature of this analysis. Is really considered a novelty instead to take all the genome-wide PRS. So they are working with SNPs identified in particular modules. And the second step, they extract GWAS effect size for those SNPs from the large schizophrenia GWAS dataset, compute the PREs for each individual cohort across the selected SNPs that we now know that they are coming from specific modules, and they test whether the TRD, in this case, the one with the polygenetic risk score, predicts schizophrenia status in external cohorts. This is the sort of validation that they use. The MVP is the Million Veteran Program database that they use for confirm the data. So in this way, they interpret the significant prediction as an evidence for each one of the TRD modules that they found. In this case, for the figure 1, they focus on the TRD1. Okay, well, this is a more deeper explaining of the shaded reds and shaded blues that they are explaining for the enhancers to promoter links that I explained. And this is the figure 2 that is coming a lot from the explanation of the figure 1. So I'm going to focus on explain that they have identified, for example, here in the panel A of this figure, that is titled Schizophrenia, OCRs, and Inferred Schizophrenia Trajectories in PFC Neurons. So from here, the analysis focused on mostly in the PFC region of the brain and also in the schizophrenia. They had to remove the other brain diseases because of the lack of statistical strength, but they keep with the schizophrenia for the rest of the analysis. So here they identify or provide the number of schizophrenia open chromatin regions, but also in the panel B here, they have a heat map of enrichment p-values for brain and non-brain related GWAS traits. So here, basically, we can see here in the top that there are a block of neurons and then followed by neurons specific and then no neuron specific and share. So they also classify this in up plus down, only up, down, and they made the same split for all the categories added here. And well, here, for example, the they explain a bit of what it means, the slash and the asterisk there. So I guess that I have here a bit of that. Okay, I don't have it. Well, so these are the relevant enrichment terms found in the ontology. Most of these related with Alzheimer's disease, outings, and major depression disorder is lack of present here, but is present in neuronal specific cases. And also, they use this specification to show that in non-neuronal populations, they have a lack of enrichment here present. So that is why they focus on neuronal populations from here to the study. And for example, here in the panel C, the panel C is this one. That is the diffusion map to visualize the global similarity among samples based on the chromatin state. As explained, these are samples, and there is the shift between controls and schizophrenia cases. And what they use is to describe the trajectory organization of these neuronal chromatin states across these suitable samples. And the trajectory correlates with the module found in the TRD1 activity, but also they found genetic liability in the polygenic risk scores, and they could link these signatures to fetal states. So here also, let's say, some of the things that take my attention is for the stratification of these neuronal PFC samples, by disease, they cluster using the latent clustering method. That is one that we have been using in some of the projects that we are doing. And also in the panel E, they have the pseudotime estimation based on the diffusion coordinates explaining 99% of the variance. So we can see here these pseudo score differences in the control versus the schizophrenia. From here, I will jump to the figure 3, the last one that I'm going to present here, I guess, is one of the most relevant, just to state what is a TRD. And for example, here in this panel A, this figure is called Series Regulatory Landscape and TRDs from Hierarchical Clustering of Neuronal Schizophrenia, CRDs from prefrontal cortex. So here they're using, in the panel A, partitional heritability coefficients using the LDSC methodology for schizophrenia risk variance, calculated separately from neurons and non-neurons. So neurons are in red here, and non-neurons are in blue. But what I like from the figure, particularly, is that they, for example, add here, they separate this in blocks. For example, this is for all the OCRs identified. So the S-score here is not so expanded, but is the baseline to say that in neurons, we have higher S-scores here identified. But when they do this, for example, in the different two brain regions that they have, ACC and PFC, we can see that the neuron populations, they really separate from the controls. So they use these figures just to state and confirm that this can be visualized in the statistics if they separate in the different two regions by the two different kind of cells that they are handling. And these, for example, are outside CRDs, that they are not in the TRDs that they are studying. So we can see that as they are not part or they are not identified, so they don't show a particular pattern. So they are just mixed here. But this is like a way to confirm what is the heritability coefficient, and they want to focus on this particular neuron populations on the prefrontal cortex based in the schizophrenia. Then I will jump over here just to say that this is a heat map here in D. This heat map is the correlation network between the OCRs, the different open chromatin regions identified, and it's the way that they use to identify modules. So for example, here you have the module 1, 2, 3, 4, 5, until the module 10. And this is like colored by upregulated and downregulated OCRs using the Pearson correlation. So what they do is to identify these TRDs here, and then they tested here in the heritability coefficient, and basically they here in the x-axis they are trying the 10 modules that they identify here, and these 10 modules are measured with this score just to see what of them are suitable or they are stable for continuous analysis. So they found that the module 1, the TRD 1, it crossed this theoretical line that they cross here. We can see here that there are some kind of blue bars here. That means that they don't pass the expected threshold to continue with the analysis, or in other words, they are not stable. So they identify as stable the TRD 1, 6, and 7. And this is to continue the analysis. So this is a sort of confirmation to say, okay, we identify these modules here, but also we want to see if they pass the threshold or they are stable heritability coefficients to continue the analysis. So for the rest of the analysis, they make different kind of tests to see if they really confirm the difference between the fetal and adult stages with the data that they handle. So for example, here they extract only the TRD6. They don't use the one here, but it's in a sort of way to show that for example, closed in fetal cortical and open in schizophrenia, they have lowest scores, while in open fetal cortical, they have, in schizophrenia, they have higher scores, in a sort of way to say that they found a linkage between this transition. So this is all that I'm going to present for this talk. And just a short summary, I just want to say that that particularly what I found really useful is that others are claiming that we need to also look into these far open chromatin regions or distal chromatin regions that can be acting in a specific disease, but we cannot do it with just identify, let's say, the proximal open chromatin regions that can be acting in the regulation, but we need to go beyond and probably build networks of these open chromatin regions in order to find modules, and then look if the modules can be acting in a specific disease. That is why they use the linking disequilibrium methodology in order to try to link this. There's a lot of steps here, really, that I don't mention, but this is basically the overview of the authors, related with the workflow that the authors present. So thank you for your presence here.